All right, guys, so today we're going to also talk about DNA isolation, so preparing the DNA templates for PCR. And so the very first thing we're going to do is collect some of our own DNA. And how we're going to do that is we're going to collect cheek cells by basically using saline solution, which is just table salt, and washing it around in our mouths, just like Cecile is doing right now. So in there, we'll have a bunch of spit and a bunch of cheek cells. And so what Cecile's doing now is that she grabbed an Eppendorf tube and she is going to put 1 milliliter of her saliva into an Eppendorf tube or a microtube. So once she's done that, what we want to do is separate out the cells from the solution. And so a really easy way to do that is to just centrifuge it. But we want to make sure that our hinges are facing out. And what this does is that it will prevent our tubes from accidentally popping open in the centrifuge. And we want to make sure we spin it at a really high speed, just like it says in our protocol. So once we have centrifuge, you'll notice that we have a pellet and then our supernatant. Remember that the pellet is the solid white part at the bottom and the supernatant is the liquid. And so we have a cell pellet at the bottom and then our supernatant. So what we want to do next is we want to get rid of our liquid or our supernatant. And so we can easily do that just by dumping it out. And we want to keep the pellet. As you see, we've kept the pellet. And so then what we're going to do is we're going to resuspend our pellet. And the way we do that is by loosening it up. And what we do is that we resuspend it in the small amount of remaining liquid that is on that is still in the tube, because we can't get rid of all the saline and all the liquid. And so what you see here is that Cecile is basically just using the pipette to move it back and forth. And so our very next step is to basically grab what we call Instagene matrix from our instructor. And so just a really quick note on Instagene matrix. It's basically just a bunch of negatively charged beads. And so this is really important because these beads will basically attach themselves to anything that's positively charged, which is essentially everything except our DNA, right? Because DNA is negatively charged. And so it's not only removing everything except the DNA, but a lot of the stuff that is positively charged inside of our cheek cells is actually enzymes that break down DNA. So we're not only maintaining our sample of DNA from degrading, but we're also purifying our sample by making sure that it only has DNA. So what we're going to do is that we're going to transfer our pellet sample into 200 microliters of our Instagene matrix solution. And like I said, they basically purify / maintain our sample from degrading. And then after that, we're going to incubate it at 98 degrees Celsius and then vortex our sample again. Or not vortex, I'm so sorry, centrifuge it. And so the released DNA is now going to be in the supernatant. Okay, so in our first sample, our pellet had the DNA. In this sample, our DNA is in the supernatant, because remember, the pellet is going to be where the beads are going to be the beads, and the beads have everything that we don't want, which means that our DNA, which is not attracted to the beads because the beads are negative, and negative and negative repel each other, means that once again, the DNA is in the supernatant. And the reason I'm repeating this is because it seems like every semester students will actually get rid of the supernatant and keep the pellet, which is the opposite of what we would want to do in this step. And so we're going to go ahead and collect 20 microliters and put them into PCR tubes. And then at this point, this would be the end of our lab for today. And so we would want to keep our DNA sample preserved. And the way we do that is that we keep it on ice until everybody is ready to basically put it in a freezer in the lab. And this freezer is at -20 degrees Celsius, and this maintains our sample from degrading until our next lab period. So feel free to move on to the next video where we talk more about PCR.